Waiting for some additional people, but so hello everyone. I just want to welcome you today to the meeting. I'd like to thank you for traveling. We have folks here from the island and Sunshine Coast. I think we are expecting somebody from the interior and across the Lower Mainland. So fantastic effort that everyone's made. I'd also like to thank and acknowledge CNETs for hosting this meeting for us. They are, strongly advocating, raising awareness, providing education for our community, and it's wonderful that they're giving us a platform to meet as a community and come together and become educated on BC-centric things that are happening. So definitely want to acknowledge them there. I'd like to introduce Amanda Rice. So Amanda is from CNET. She's part of the board of directors and has joined us today here from Toronto, helping us have this meeting, leading the technical aspects of the meeting. And most importantly, I'd like to introduce our two speakers today. So Dr. François Bénard is he is a BC Leadership Chair in Functional Cancer Imaging with BC Cancer Agency and UBC. Did I get that right?
Yes.
Yes. So welcome. And Dr. Don Wilson is a nuclear medicine physician and radiation oncologist with the BC Cancer Agency. Welcome. So just a quick review of what we're going to do today. We're going to go through presentations. We'll have Q&A. We invite you to join us for lunch. It'll be served about 1 o'clock today. And that's it. So, any questions or, yeah? Okay, fantastic, thank you. I'll turn it over to you too.
Oh, thank you.
This one? The other one.
So first of all, thank you for having us and welcome to Vancouver for those who are outside, but thanks for coming for the invitation and agreeing to come and meet with us. I must apologize in advance. There's a risk I might start a coughing fit anytime. I'm just recovering from a cold, and Don is just starting his cold. So, doctors don't always take good care of themselves, but I hope we're fully loaded enough on cold medication to be able to go through without coughing too much. So we've decided to split our presentation. I'll be talking about some of the technologies that are used to diagnose and localize neuroendocrine tumors. And Don will talk about therapy, and in a minute, through that process, we'll explain a little bit of what we're doing in BC and what our program is, and what our objectives are to bring new diagnostic tools and new therapeutic approaches to help people with neuroendocrine tumors. So I'm an imaging guy where we do both imaging and therapy in nuclear medicine. In the past few years, I've been mostly focusing on developing new imaging methods. So I'm a clinician scientist. That means I spend part of my time doing clinical care, but in my current workload, it's mostly doing diagnosis and doing PET scans, and also spend a large part of my effort doing research. And some of that has been directed towards neuroendocrine tumor, and I'll give some examples of what we do. And the type of imaging we do is called molecular imaging. So it's a term that's been used to coin the fact that we're trying to really visualize the presence of specific abnormalities on cancer cells. So we're not looking at changes in the anatomy or structure of organs. We're really looking at specific abnormalities that are found in cancer cells in order to use those to detect cancer and find out whether cancers express specific features that we can use to treat them. So there's typically molecular imaging methods use specific probes so that we inject intravenously, so it's just a small injection, and these are drugs that are engineered to really home in on cancer cells and accumulate in large concentration in cancer cells. And then you need to have a signal that you can detect by an external imaging system. And really what's practical for use in clinical care is really radioactive isotopes. So we're all scared about radiation because it's known that radiations in large quantities cause cancer, and we work with the assumption that any kind of radiation is harmful, but it's really dose related. It's like smoking. If you smoke 1 cigarette, the risk is minimal. If you smoke 2 packs of cigarettes per day, then it's a bigger risk. So we keep for diagnostic testing the dose of radiation very low. But the advantages of using radiation is we can see deeply in tissues. If we were to use light, for example, we can't see the skin absorbs light. Even a few microns of, not even a millimeter of skin thickness would block light. So this is the only way we can use probes that will give us a signal that we can take images. So the typical dose of a scan that we do is like a CT scan of the abdomen or a CT scan of the chest. There's the risk of cancer related to one scan is extraordinarily minimal. We all have about 40 to 45% chance of getting cancer in our lifetime. The risk of a cancer estimated by one diagnostic scan or one CT scan, for example, is 0.0001%. So it's not meaningful in terms of the risk that we spontaneously have of developing cancer. Just to explain a little bit how this works. In order to be able to do our scans, our typical day in a nuclear medicine department starts by the production of the radioisotopes we need, and we use a machine called the cyclotron to do that. And that's in the upper left corner, the blue machine. This is actually a BC invention, built in BC, made in Richmond. It's the results of a research program that was done at Triumph that started a big national lab for physics on the UBC campus. Then we transfer our radioactivity to a room where it looks like a series of stainless steel fridges, but these are essentially large boxes with lead because we produce a large quantity, and the staff that works with them day in, day out can't be exposed to a dose that would be enough to do scans in 50 different patients. So we need to protect the staff who work with this, so we actually handle the radioactivity behind these big units. And then there are small robots that do that, or small devices that do that automatically. It's a little bit like a bread machine that will do your bread automatically. At the end, you end up with your product. It's monitored by staff because these aren't quite as reliable or as a coffee machine or a bread machine. It's a little bit more complicated. And at the end, we end up with a product that we can inject in people and scan them. And nowadays, we combine scans, so we have both the functional images, like this one on this side, and the anatomical images. In this case, it can either be this is an MRI scan, but it could be, usually we use a CT scan, and we combine both images to get the benefits of both having the traditional scans and having the molecular imaging information from the drug, the radioactive drug we inject in people. So I checked the numbers. We have over 60,000 patients who underwent PET-CT scans in British Columbia since 2005. So Don started the program in 2005. I joined in 2008. And we have a busy clinical program, and we do scans to find out whether somebody has cancer or not, whether if somebody's been diagnosed with cancer, whether the cancer is operable, whether they've responded to chemotherapy, and if they see abnormalities on CT scan, is it still viable tumor, or is it just fibrotic tissue that's not likely to go back? And so it's helping a lot of people, and we do those scans for a lot of many common cancers: lung cancer, colorectal cancers, cancers of the lymph nodes, and so on. And what we've noticed with this is we've significantly reduced, for example, the number of radiation therapy in patients with cancers of the lymph nodes. It avoids a lot of unnecessary surgeries in patients in whom they want to do surgery, and then we find out the disease is more extensive than they thought initially. Generally clearer diagnosis and better guidance for radiation therapy planning. We also have a research program that we've built up, and in order to be able to test new compounds that might improve diagnosis, we need to be able to do this. We can't go directly from the chemistry to inject things in patients. So we need to have models that we can test and make sure that this works before we go to patients. So we use the smaller versions of the PET scans and the SPECT scans that you'll see in nuclear medicine departments to be able to do things in mice so that we can take a step to make sure things work before we end up introducing them in the clinic. And so we have a research program where we're developing a number of applications for different types of cancers, and that includes, for example, melanoma, a skin cancer. So this is essentially, this is a mouse, but it's a CT scan of a mouse. This is why you see the skeleton. So we have a CT scan and a PET scan. It could be a patient. These are small patients. So we take very good care of the mice and make sure that this is done very humanely. But it allows us, for example, this is something that targets melanoma. This is another probe that targets breast cancer. Another one that targets breast and prostate cancer. So we have a whole series of new imaging agents and therapeutic agents that we develop for several tumor types And we bring them then to the clinic where we can test different imaging agents. So this is a series of examples of images from patients that participate in some of our clinical trials. It's not particularly relevant for today's scans, which why we're doing those particular projects, but this is just to highlight the fact that we have an extensive research program really to develop some of these new imaging probes that are used to detect cancer. Now, there are different types of probes that we use, and I'm introducing this really to explain what we can do for what's been developed by others and what we can do for people who have had neuroendocrine tumors. So some of, we call them radiolabeled small molecules. So these are small compounds, a little bit like drug or Tylenol or, that will bind to a specific function in the cell. Sometimes it's based on a natural compound that our body makes and is used for various body functions. And then they're modified. Sometimes it's based on drugs that have been developed by pharma to affect a certain pathway related to cancer. So we can tag the small radioactive isotopes and use those as imaging agents. We can also use antibodies. Just like our body, if we have a virus, we start producing antibodies, and the antibodies that are produced are very, very specific to the virus. We can also develop antibodies against cancer-related proteins and use those as imaging or therapeutic agents. So there's been some examples of successful ones that have been used, for example, to treat cancers of lymph nodes. And then we can use peptides. So peptides are compounds that are made of amino acids. So we are all made of proteins. Amino acids are the little building blocks of a protein. So a peptide typically will have 5 to 20 little amino acids stuck together. And they're very good at recognizing specific proteins that exist in cancer cells. And those are very convenient to work with because it's very simple to make them. It's very simple to attach to radioactive isotopes. And there's a number of cancers that can be targeted using small peptides. There's challenges with working with them, but the best example of a peptide that's been developed is really related for neuroendocrine tumor imaging, and I'll explain why this works. And now we need once we have a molecule that we know will bind to cancer cells, we need to attach it to radioactive isotopes. So this is another complicated field because there's lots of radioactive isotopes. So a molecule, an atom, will become radioactive when it's unstable. So in nature we have mostly stable isotopes because most of the radioactive ones disappeared billions of years ago when the universe was created, and only the stable ones remain. So it's unstable because the nucleus has an unbalance between its positive and negative charge. So in order to become balanced, it needs typically to expel the charge. So this can occur through several processes. And I won't go in details, but some of them just expel energy in the form of a ray that we can use to detect. So this is what is used for technetium-99, which is one of the most commonly used radioactive isotopes for nuclear medicine imaging, or typical bone scans or cardiac scans that people go through in hospitals or use this isotope. Another one is called electron capture, and this is what is used for iodine-123 and indium-111. So these are common diagnostic isotopes that are used. Another decay mode is called positron emission, where instead of one ray, 2 rays are emitted 180 degrees apart. And this allows us to really create much more precise images compared to the other ones. And this is an example comparing what is a standard bone scan compared to what is a PET scan. So the one on the left is a single photon emitter, we call, so it emits 1 photon. And the other is a positron emitter, so that emits 2 photons. In one case, we need to put a heavy lens. It's like when you take a picture with a camera, but it's made of lead. So it blocks out most of the light to know where it's coming from. And the other one, because the rays are exactly 180 degrees apart, we have machines that can localize very precisely where this is coming from. And it creates sharper images, and it requires less radioactivity to produce an excellent image. So you can see much smaller lesions using a PET scan than you can do using a conventional single photon scan or a conventional nuclear medicine camera. So this is really why PET scans have become so useful, because it detects a much smaller lesion at a much earlier stage. Now there's also radioactive isotopes that are used not for diagnosis but for therapy. And the main difference is that instead of emitting a ray that doesn't interact with the patient and goes out of the patient, those emit particles that deposit their energy locally. So and depending on the type of isotopes, it can have a small, very, very small particle or very, very heavy particles that deposit way more energy. So the most potent ones are called alpha emitters, but the most available ones are called beta emitters. So lutetium is a beta emitter. It emits a small particle typically move up to 1 centimeter away from where it's bound, and will hit targets on the way on its track and create damage there to kill tumor cells. The alpha emitters will have a much shorter range, but it's a big cannonball. Will just stop and deliver its energy very locally. That's why they're much more potent, because it remains more closer to where the tumor is, and it also delivers more energy. They're also very, very difficult to get. So there's a number of isotopes that have been used to tag peptides that are used for imaging, and I'm explaining this because you'll see in the types of scans that can be recommended by doctors that they use these isotopes. So technetium-99m is the one that's used for bone scans and a lot of common nuclear medicine scans. It has a half-life. A half-life means because it's unstable, it disappears spontaneously. So every 6 hours, you lose half of it. Indium-111 is used primarily for neuroendocrine tumor imaging. So a nucleotide scan that some people may have had in hospitals uses indium-111. So it's got a long half-life. Typically, the images are done after 4 hours, and then people have to come back the next day. And it's been for many years the mainstay of what we had available to diagnose neuroendocrine tumors. However, it has a hard time seeing lesions that are smaller than 2 centimeters. 2 centimeters is pretty small, but for cancer imaging now that's too big. We want to go smaller than that. Gallium-68 is an isotope that's been very, very popular in Europe in the past 5-6 years. And I'll be showing some images. This is really what's changed the landscape for neuroendocrine tumor imaging. And fluorine-18 is another isotope. It's only got a 2-hour half-life, but this we use widely because we make very, very large quantities of that every day, twice a day at the BC Cancer Agency for cancer imaging. So F18 is the most convenient one to use because we have plenty of it available, and it's not easy to make, but now the technology is so mature that we can make this on a routine basis. The problem with gallium is it's got a much shorter half-life, and when we only have small amounts of it, so when we make a batch to make a new compound we can do 1 or 2 patient doses, and then we have to start again for another 1 or 2 patients. With fluorine-18, we make so much that we can have a full day's worth of scans essentially from 1 production batch. So it's much more expensive to use gallium than F18. And it's also, because the gallium is getting popular all around the world, there's a shortage problem that's about to occur with this isotope. It's getting tougher and tougher to get access to it. So I know I'm talking to a fairly educated crowd about neuroendocrine tumors, but I just thought I'd summarize a little bit what these tumors are. So they're tumors that grow from neuroendocrine cells that occur everywhere in the body. The majority of those come from cells that are called enterochromaffin cells. So entero means it comes from the gut. Chromaffin means that the dyes that the pathologists were using had some chrome in it, and they were specific for those cells, and that's how they discovered that these cancers came from different cells than other cancers. And they can originate commonly from the gut, sometimes from the lungs, often from the pancreas as well, and they have a very wide range of behaviors. Some tumors are very aggressive. Most are slower growing than other types of cancers, and people can have this for years with big symptoms before they're diagnosed. And that's often a tragedy with those tumors, is because people have had them for a few years without knowing they had them. The symptoms are not specific, and often it's diagnosed late, and the cancer is spread elsewhere. And even if you resect it, it's by the time it causes symptoms, it's fairly late in the game. They often secrete hormones that cause symptoms, because these cells and the cells that cause the tumor are used to regulate, for example, the motility of the gut, and they have local function, and so they secrete a lot of substances that can cause a lot of symptoms. But it also means there's a lot of regulatory mechanism on those cells that make them active or inactive, which we can use to target the cancers. And there's about 2.5 to 5 new cases per 100,000 people. And the incidence has been increasing since the 70s, probably because awareness and the diagnostic techniques have improved. This cancer has always been there, but we just diagnose it more often than earlier than before. And there's about 35 people per 100,000, and probably higher than that, who have cancers. Probably a lot of people have very, very slow growing versions of those cancers without knowing they have it. These tumors are graded as well based on how quickly they divide. So there's a series of pathologists, when he looks at the slide, can find markers and identify which grade of tumors this is. So they're generally separated in low grade, intermediate grade, and high grade. Essentially, this means that high grade tumors divide more quickly and they grow more quickly than low grade tumors. So the treatment is often determined based on the types of grade that somebody has with those tumors. It's also a very complicated landscape because it's a broad category of tumors, neuroendocrine tumors, and they're very, very different in their behaviors. The most common ones are called carcinoids, and they come from the pancreas, the gut, or the lungs. There's also functional pancreatic tumors that secrete hormones. For example, insulinomas will secrete insulin, which is a hormone that drives down the blood sugar. So people who have those tumors, their blood sugar drops and they faint and pass out. Sometimes even happen in newborn babies, for example, where they have a tumor in the pancreas when they're born, and then with the sugar level, they have a hard time keeping it normal. And there's other tumors, neuroblastomas that occur in babies. Tumors of the adrenal glands. So these are all considered neuroendocrine tumors, but they're treated very differently from carcinoid tumors and so on. So it's a wide group of different tumors that are lumped together when we talk about NETs, and they each have different treatments. So the conventional imaging methods that are used is ultrasound, CT scans, and MRI scans. Those look at the anatomy. So you have to have a mass that's sufficiently large for the tumor to be detectable. What's been used for many years is a peptide that targets the many neuroendocrine tumors called indium-111. The actual name is pentytriotide. So the pharmaceutical companies give these weird names to the compounds, but it targets neuroendocrine tumor cells based on the receptor called the somatostatin receptor. So it's an analog of a natural hormone that we have that's expressed in the tumor cells, and they've tagged it with a isotope called indium-111. So those scans cost anywhere from $1,000 to $1,500, and they take a couple of days to scan. It's slow. If any of you have had those scans, it takes a long time. It's about an hour of imaging, and then you need to come back the next day. But it's been very useful to detect and see whether the cancer is spreading or where it's spread. However, the problem with it is that it misses small tumors, and sometimes more aggressive tumors might be missed by this kind of imaging tool. There's another compound called MIBG, which is used for tumors of the adrenal glands and some types of neuroendocrine thyroid cancers. I won't be talking about this today. So what's the somatostatin receptor? Because this is what we're mostly talking about today. So it's a receptor is when a cancer cell has a nucleus and it's got a membrane around, and that controls what kind of substances get in the cell. And there's a lot of protein. So it's made of fat, the outer layer of a cell. And there's proteins that give the cell structure and solidity, but a lot of those proteins have specific functions to control how a cell behaves. And the somatostatin receptor is one such protein that's present at the cells. Because it's on the outer layer of the cell, it's accessible. If you give an IV drug, it can bind to that receptor. And it's expressed in many tumors, neuroendocrine tumors as well as non-neuroendocrine tumors. And especially expressed in those tumors that grow slowly or moderately slowly. Typically, the very, very poorly differentiated aggressive tumors, which are fortunately more rare, don't express large quantities of those receptors. The particularity of this receptor is when you stimulate it, when you have a compound that binds to it and activates it, it slows down the secretion of hormones and it slows down the growth of that tumor, which is why you have octreotide, long-acting octreotide, and so on are used to treat mostly to treat the hormone secretion syndromes, but they're also used sometimes to slow down the growth of the tumors, although there's less evidence that this really is that effective, but it mostly controls the symptoms very well. So long-acting somatostatin works this way. People get their monthly injections, and then it controls the symptoms and hopefully slows down the growth of the tumor. And there are 5 subtypes of somatostatin receptors. They have different functions in the body. The most common one that's expressed in the tumors is a somatostatin type 2. So this is because it's highly expressed. It's very present in large quantities of the surface of the cancer cell. This is what's being used to target as a target for imaging as well as radionuclide therapy. So currently what's used in the clinic is the indium-111 compound that's available in all nuclear medicine departments in British Columbia and throughout Canada. In Europe, some companies have developed compounds labeled with technetium, so it's easier to work with, the images are slightly sharper, and it's a one-day imaging session. However, those compounds were never approved for use in Canada just because it's a small number of patients relative to the cost of filing approval, and there was a Montreal-based company that had a good compound, and it was developed in Europe but never in Canada, just because of the cost of getting to market. There was an available product and so on. But in a few groups, especially in Germany, started to develop PET imaging analogs, so with the positron emitters of these compounds. And there were new generations of compounds that were developed after these first few were developed. So they bind more tightly to the tumor cells, and they were putting PET isotopes with them. And they started using it in their clinic, and there's a few clinics in Germany that started to collect large number of patients, and this grew in Europe quite a bit. No commercial sponsor. These are centers like ours that decided to use the compounds, and there were patents on those compounds, but at least the pharmaceutical companies did not prosecute the centers and say you can't use it because you lost money. So they let them do it. But it became very, very popular because people were doing clinical studies and they realized, oh my God, this is much, much better than what we've been using. So you'll see some of those names that have been used. So there's DOTATOC, DOTANOC, DOTATATE. There are 3 different compounds that are almost identical with very subtle difference in their chemical structures. They've also been labeled with a variety of different isotopes, and I'll show you some examples that we developed in our lab using F-18. But essentially, they all work well, but there's small differences between the different compounds. There's also other imaging agents that can be used. So the FDG is a sugar analog. So this is a kind of sugar that we use to diagnose cancers every day in our center. We're open today. Both of us are reading scans the whole day. When we finish this meeting, we go back to work. So 6 days a week in BC, we do scans using this compound. And it doesn't work in some neuroendocrine tumors, and it works in others. So it's almost a mirror image of the peptide imaging. So the FDG scans are good for the fast-growing aggressive tumors, but it doesn't work well for the slower-growing ones, while the peptide imaging is better for the slower, mid-growing ones. There's also a scan called fluorodopa. So this is an agent that was used for brain imaging to study Parkinson's disease. So we have it in BC because it was being used for years for research studies on Parkinson's disease. But it's a precursor of some of the hormones that are secreted by many of those tumors, and it's actually quite good at diagnosing neuroendocrine tumors. And there's other compounds that are being used in other centers. You may see 5-HTP, which is used in 1 or 2 centers in Europe for neuroendocrine tumors. That works a little bit like F-DOPA. And it depends on the types of very specific subtypes of tumors. So these are used for very, very rare tumors. So I'll skip just briefly, just to show you some examples about F-DOPA. So this we've done, 250 patients perhaps in BC for many years. And essentially you see here, this is part of the brain. So this is what controls tremor and movement, which is why it was used for Parkinson's disease. And this is the liver of a person, and this is the bladder, and you see the pancreas. So these 2 dots here represent tumors that are coming back. So we've been using this very successfully because we had it available with our partners at the Big Physics Lab Triumph, which was making these isotopes for the Parkinson's project. And we've used it to diagnose tumors. It's particularly good for a type of tumor, neuroendocrine tumor of the thyroid gland called medullary thyroid carcinoma. And this is just an example comparing an FDG scan where you see essentially very minimal or no tumor, and you see on the left with F-DOPA, where you see a lot of all these dark spots here. These are all in the liver. These are disease that could not be seen by one scan, but can be seen by the other scan. So it always makes us nervous when we start to see things with one method and not the other. It's as a famous American Secretary of Defense said once, we don't know what we don't know. So it's always good to have multiple imaging methods to look at the same problem. Now, F-DOPA, unfortunately, we've had to put a hold because it's unavailable currently. There's over a year's worth of work to modernize the synthesis production. So we have a larger backlog of people who need imaging for neuroendocrine tumor. This was not made by our group. We were relying on an external group to make the compound for us, and they've had to stop their production for numerous reasons. So in the meantime, we decided we would bring the gallium compound so that we can offer the service to the patients. And the reason this is needed is you see those two images. This is the same patient, and those two is an anterior and posterior. So you're looking at the person from the back, and this is from the front, which is why this tumor here, you see it more clearly there. So this is a conventional Indian scan done in the nuclear medicine department. On the other side is a PET scan. And obviously you see way more disease with PET scan compared to the Indian scan. So it's much more precise. It's also faster to do the scan and more comfortable for people. So the advantages of the gallium over the indium is that it's better to detect small lesion. You inject the compound and you image 1 hour afterwards instead of doing it 4 and 24 hours. And it also takes less time to do the images, and it's also less costly overall to do. So in summary, it's better, faster, and cheaper. So there's no question that the gallium is the way to go for the future. So there's all these three compounds that are there, and I have colleagues in Europe who swear by one and say the others are not as good, and others will say something different about the three. The bottom line is that they all seem to work well. They all have subtle differences. None of them are approved for use in Canada, so we have to use them in what's called a clinical trial. It means a controlled way in which we can make sure the agent is safe, track its efficacy. So we have to get people to sign a consent form and participate in a research project in order to be able to bring this to Canada. It was just approved in the US because a commercial company picked this up and decided to invest in getting this approved in the United States. Our goal is to do this and get it approved just like we had the FDG approved by Health Canada, and do this but in the public sector. So there's some comparison between different ones. As you can see, in most studies, when you've compared the compounds, isotopes has been slightly better than the other ones because it sees small liver tumors better. So this is the same patient. A group of colleagues a few years ago compared both of them and thought that the isotopes was slightly better. There's another one called the DOTANOC, and it was compared to dotatate, and in this case, dotatate was a little bit better. So my impression, and these are small studies, so they're not large studies where we can unequivocally say one is better than the other, but we've come to the conclusion probably the best one to bring to BC was DOTATOC, because when it was compared to the other two, it always seemed to come out a little better. That doesn't mean that the other types of scans are not good, it's just that the results, the number of lesions seem to be a little bit better with the DOTATOC. And this is an example in some neuroendocrine tumors. It also shows how variable things are. Sometimes even within the same patient, tumors can behave in a different way. The image on the left is a F-DOPA scan, which we've relied upon and is a very good imaging agent, but the one is the same patient with in this case, DOTANOC, but somatostatin receptor imaging, and you can see that there's a lot more disease seen there. And it changes in agent, because if you think there's a single lesion, then you might consider surgery, or you might consider, local forms of therapy. Well, if you know it's spread, that means you're just wasting people's time and creating, delays rather than giving them the appropriate treatment. So you may also hear something called about agonists and antagonists, because this is a relatively recent development as well. So when we are talking about receptors, an agonist is a term that's used to say that something that stimulates the protein. An antagonist is what blocks it. So people were using agonists because an agonist slows down the growth of the tumor in the case of a somatostatin receptor. Most drugs are actually antagonists, they block the function. But this one was, we use the somatostatin analogs as an agonist because they slow down the tumor growth. So nobody really looked at antagonists in the context of neuroendocrine tumors until when somebody labeled it, tagged it with an isotope, and realized that they see much better the tumors with it. And there's complicated reason why this is, but there's a series of new compounds being developed which are antagonists, and it's specifically only for radionuclide treatment because it binds to more proteins at the cell surface, and you can get apparently a better signal. So we've tried comparing in our lab, but we don't see any difference between the agonist and antagonist. Other labs claim that the antagonists are better. I'm still a little bit skeptical, but there's certainly new drugs coming in that may be better than the agonists that will come on the horizon. However, we are going with the agonist because this is what the large-scale studies have shown would bring benefits for both diagnosis and treatment. The antagonist is still a little bit further back, and our lab data doesn't quite agree with what's been said by others. So we're still on waiting a little bit about this. So this is an example from a group in Germany where they compared the standard octreotide with an antagonist, and the advantage of the antagonist is that it had lower uptake in normal organs, so they could see tumors better. And they also believe that by giving the antagonist, there might be fewer side effects related to treatment because normal organs receive less radiation. So this is still an evolving story. But if you hear there's a compound called GR11 or OPS-202, these are two of the new compounds that are starting to be to enter clinical trial. So one of the companies that provides long-acting somatostatin is called Ipsen, and they've purchased the rights for this and are looking into whether this is an advantageous treatment or whether it's better. So there's still developments in terms of bringing new treatments that may be more effective even than the current ones. I'm sorry for showing mouse images. Some people find those creepy. It's not a skeleton of a mouse. It's a CT scan of a mouse. It's like an X-ray of a mouse. So those are different versions of the compound from the antagonist to the agonist that we've tagged with different isotopes. We've made a gallium version, so it's much faster to do things in the lab when we don't have to go through Health Canada to get them approved to use them in patients. But in this case, the advantage is that we've developed a technology to tag the peptide with fluorine 18, which is what we use commonly to scan cancer patients. And this we can make very, very large batches. So for when we're looking at an entire province like BC, it makes it much more attractive because we can, get people and come in once a week instead of doing 1 or 2 patients with one batch, and we can do 5 or 10 patients with one production batch. It's also better. The compound, the way we've attached the radioactive isotopes, makes the compound bind much more tightly to the proteins. So this is the one on the left, the TATE. We've compared the TATE to TOC in the GR11, the agonist and the antagonist. And the version of octreotide labeled with F18, at least in the mice, are much better. So this is what there will be a clinical trial where we'll be testing this and bringing this to patients. We're in the process of ensuring that there's no toxicity associated and it's safe to use. So that will take a few more weeks or months. And we're hoping that in the first half of next year, we'll have this available for a clinical trial. We're still pursuing with the gallium-68 because that's what's validated, and we'll need to compare it with the F18, so we'll have both available for a period of time. And if we find that one is better than the other, we'll stick with the better one to offer patients. So what are our current plans? We've been working for a long time with bringing the gallium-68 imaging. It's easy to make the compound. It's not so easy to make it compliant with Health Canada regulations. And that's where most of the effort is to document and do the production. So I was hoping that by this time this year we would have the gallium-68 available. We've had delays for multiple reasons. Things never quite go as fast as I would like them to go. But right now we're hoping to have this by second quarter of 2018. We'll probably have the F18 available at the same time. But this will be a much smaller study. So the gallium, there's a lot more data, so we can do a broader, more open study to include as many people as possible so that anybody who needs a scan can be eligible to get one. And the F18 will probably be a subset of people that will offer to participate. And radionuclide therapy, we're working hard in parallel, and then we'll be talking about this. We're hoping that by this time next year, our program will be up and running. We are doing research on the benefit of alpha emitters. If you recall, I was talking about those isotopes that emit these strong, these big balls that are much more effective at killing tumor cells. So just yesterday it was announced we received $10 million in funding to build the infrastructure at the big physics lab in Triumph that will potentially more than double the production of alpha emitters in the world, right in our backyard. So we will have these isotopes available as well to bring to clinical trials. And I believe Don may have some images comparing the efficacy of those compounds, but there's only been one small clinical trial with neuroendocrine tumor imaging in Europe. To do that trial, they had to scavenge probably all of the alpha emitters they could find in Europe at the time, but it showed even people who were not benefiting from the TCM therapy were benefiting from the alpha emitter therapy, so that's yet another line of treatment available. And of course, there's a lot of questions about whether this can be combined with other treatments. Right now, it's used as standalone treatments. There's a group in Australia that are combining oral chemotherapy with radionuclide therapy and are finding that for the more aggressive neuroendocrine tumors, this works very well. So this essentially highlights some of our points. Thank you. This is a clinic colleague, so we'll have to pick it up. Sandra, did you want to do questions right now, or did you want to wait until the end?
I'll put that out to the group. Do you have any specific questions you'd like to ask now, or would you like to wait to the end of the presentation and ask everything at once? Okay, I think we're going to ask everything at once.
Perfect. Thank you. So I'll just have to step out to take the call, and I'll come back and hand over to Don.
Thanks very much. Thanks very much to the organizers for inviting us today. Thanks to CNES for the great work that you folks do. And thanks to everyone else here for coming and spending some time with us here today. What I was going to do is talk about a program that we're very excited about. It's something new for BC, and something I think that will benefit a large group of patients with neuroendocrine tumors. And so for us, this is a launch basically of a new therapy initiative. This is a little bit of a background. I think you folks probably know this, but obviously neuroendocrine tumors are a heterogeneous group of tumors that arise from neural and endocrine cells of neurocrest origin. Probably about 70% or so arise from the GI tract, about 20% from the bronchial tracts, and then there's a small number arising from things like medulloblastoma, pheochromocytoma, medullary carcinoma of the thyroid gland, etc. So it's a very large, very diverse group of tumors. And as François mentioned, the incidence has been increasing over the past 30 years. Part of that maybe we're maybe a bit better able to detect these tumors, but nonetheless we are seeing an increased incidence. We see about 200 new cases per year here in BC. And one of the problems with the disease is the fact that it doesn't often present until it's quite advanced, so you don't have symptoms to warn you that there's something wrong until sometimes it can be quite advanced disease. So probably more than half of patients actually present with advanced stage of disease. The treatment of neuroendocrine tumors also varies quite a bit, and it's very individualized, and it depends on the stage of the disease. So if it's picked up early when it's localized, it can be cured with surgery. If it's spread beyond the local area into adjacent organs or tissues, then other local therapies such as surgical debulking, ablation, embolization can sometimes help with symptom relief. And there's some systemic modalities as well that can prolong survival. And these include therapies such as the short and long-acting somatostatin analogs. There's various chemotherapy agents that can be utilized. And there's some biologic agents as well that target specific biochemical pathways in tumors. So the there's quite a broad range of treatments and different options depending on the stage and the aggressiveness of the tumor. One of the newer therapies out there is peptide receptor radionuclide therapy. So one of the things you've probably noticed in this field is there's a lot of acronyms and PRRT and DOTA and DOTA and so on. It's I understand it can be kind of confusing, even for us sometimes it's a bit confusing. But anyway, one of the unique features of the neuroendocrine tumors is the overexpression of somatostatin receptors. And this is something we can take advantage of with PRRT. So what we have is basically a radionuclide, like here for imaging we would use gallium-68. And then we link that to a peptide that is designed to bind to the somatostatin receptor. So what we have in effect is like a smart bomb or something that's going to go and go through the bloodstream, but it will bind specifically to these receptors on the surface of the neuroendocrine tumor cell. And it's sort of like a lock and key mechanism. So once it binds, it sticks quite firmly there. And we can take advantage of that with our agents for imaging and for therapy. So when we talk about therapy, what we'll be utilizing here is lutetium-177. So this is what we call a medium energy beta emitter. So it emits electrons or small particles of energy as it decays. And these little particles can travel a distance of about 2 millimeters. And it's over that 2 millimeter range where it interacts with the materials in the tumor cell and causes irreparable damage that leads to the death of the tumor cell. The lutetium has a half-life of about 6.7 days. So that means every week or so you have about 50% less of the radioactivity remaining. Also, that can be a good thing too, because that means when the DOTA K binds to the receptor on the tumor cell, it can stay there for a few days and emit radiation. So it's not like it's over in an instant. It can be ongoing for a period of time. The other nice thing about lutetium is that it also emits gamma rays. So these are like X-rays. And these X-rays we can actually detect with gamma cameras. So the beta particles, they only go a couple of millimeters, so we aren't going to be able to detect those. But these X-rays will actually exit the body, so we can actually image where the therapy molecule is in the body. And that helps us understand where it's going. And we can also use it to calculate the dose of the need for the treatment. So this is an example here. This is the same patient, and this patient has a large neuroendocrine tumor mass in the liver. This is on the CT scan. You can see it's a low density area compared to the normal liver. So part of that is probably necrotic tumor. But around the periphery, there's some active tumor cells. So this is with the gallium-68 imaging agent. And this is a PET-CT scan of that patient. You can see that this red stuff here is the activity sort of in the viable tumor cells around that larger cavity. And with the same patient, after receiving lutetium dotatate injection for therapy, we can see the same pattern of uptake on our SPECT cameras. So you can see that, again, that rim of high activity around the necrotic part of the tumor. So that's telling us that the therapy is indeed to basically bind to the same receptors that we're imaging with the gallium-68 dotatate. So what happens then is we talked about having the radioisotope bound to the peptides. So this thing called a chelator is what binds the isotope to the peptide. And it's this peptide which is designed to bind to the somatostatin receptor. So we manufacture the lutetium dotatate, this here, and then we inject that into the bloodstream. It circulates throughout the body, and then it will bind to these somatostatin receptors on the surface of the tumor cell. And like I mentioned, it's a very tight binding. Once it's bound, it becomes internalized, so it goes inside the tumor cell. And then over a period of time, you get more and more of the lutetium dotatate molecules binding to the more and more of the somatostatin receptors, and you get more and more of the radioisotope internalized inside the tumor cell. And then we get more radiation inside the tumor cell. And what's really key to killing the tumor cell is to irradiate the nucleus of the tumor. So that's where the genetic material that the tumor needs to survive is located. And what happens is that the DNA of the tumor is like a double helix. And what happens is the particles, those beta particles, they cause breaks in the DNA that the tumor cannot repair. And so what happens then when the tumor tries to make proteins, or if it tries to divide, it's unable to do so and it basically dies. And so that's how it works. It's really like a targeted therapy for this type of tumor cell. So people have been trying or using radiolabeled somatostatin analogs for therapy for now over 20 years. And there's been small studies with fairly small groups of patients showing us that there actually can be favorable responses. So some studies, small studies, have shown response rates up to 46% compared to standard therapies alone, and also show improved quality of life in a lot of these patients. So some promising work, but fairly small studies. And this is an example here of how effective it actually can be. So this is a patient at baseline. So this is a gallium-68 scan. And you can see multiple tumor deposits sort of throughout the body. And then after therapy with the lutetium-177 dotatate, you can see that most of those lesions have completely disappeared. There's still a few around, but substantial improvement. So I think it's important also to realize that where we're at right now, we're not talking about curing patients with advanced disease, but we can make I think a big difference in terms of symptoms and progression-free survival and overall survival, hopefully, as well. So it's promising results. And what really got us excited, was this one study here that was published in January of this year in the New England Journal of Medicine, which is a very prestigious medical journal. And this was it's called the Netter study. And some of you may have heard about this. This was an international multi-center phase 3 trial. So it involved 41 different medical centers in 8 different countries. And it was a phase 3 trial, which means it's a randomized study. So in the scientific world, this is like one of the most powerful types of scientific studies that you can do. So when we get data from the phase 3 trial, it's very robust. And in this trial, they studied 229 patients. They all had well-differentiated metastatic mid-gut neuroendocrine tumors. And what it showed is that there was a much better progression-free survival in the patients that were treated with the lutetium inhibitor. So the top line here, this green line, is the patients who were treated with lutetium, and this purple line here was the control line. So these were patients who received the triotide long-acting. And you can see the patients with the lutetium did much better. For the control arm, the median progression-free survival was 8.4 months, and for the lutetium, it was estimated to be about 40 months. So a significant improvement in time to progression for the lutetium-treated patients. And at the time they published, there was also a trend to improve